So hi, everybody. I am so grateful for the chance to speak today. And I really am I'm hoping to be able to go to the next WAGR weekend in person because I miss you all very much. So my journey with IWSA goes all the way back to when I was first studying about WAGR syndrome as a medical resident, but I had never actually met any patients. And it was when I started 20 years ago at the National Institutes of Health, I decided I really wanted to understand the genetics of endocrine disorders. And I wanted to study WAGR syndrome because of the description of the many symptoms that patients have. And so thank goodness for Google, I was able to find IWSA, and I was able to go to my first WAGR weekend in 2006 at Levittown, Pennsylvania. And then after that, I launched into enrolling patients to come to the National Institutes of Health in Bethesda, Maryland, to study phenotype-genotype correlations. And so at the time, a lot of people already knew kind of basics, and that was what I learned in residency, which was that Wilms tumor and the genital urinary anomalies were due to a particular gene called WT1, and that the aniridia, as Kelly reviewed in her talk, is due to PAX6. But the range of developmental differences was not really well understood at that time, and so kind of question mark there. And then there were a few other additional features that people started exploring. So people found that you can get osteochondromas if you have a deletion boundary that's big enough to encompass EXT2, and that's a picture of an X-ray that shows that. And then if you have a deletion that's big enough to even include ALX4, then you get these holes in the skull called parietal foramina. And so those were pretty much the things that were kind of known at the time when I first started studying WAGR syndrome. And the reason why I was interested is because endocrinology is the study of hormones. And we now know that hormones are a really big part of what's a person's body weight and their appetite. And it also turns out that hormones and neurotransmitters are really important for pain perception too. So those all kind of got bundled together as big question marks based on some of the symptoms that people have for WAGR syndrome. And the gene that I was really interested in was brain-derived neurotrophic factor to try to tie all this together. And the reason is because at that point, people knew just a little bit about BDNF. I won't read this whole slide, but it's basically just that it's a really important protein for the way the brain functions and the way the neurons work. And this basically led to questions about, okay, well, what happens if there's something different about BDNF? So when people looked at mice, they found out, well, if a mouse has something different about its BDNF, in this case, one copy of its BDNF gene, because everyone has two copies of each gene, if one of the two copies got deleted, what would happen? So you can see that that mouse there on the left weighs a lot more than the mouse on the right. And it turns out that that mouse is really hungry too. So that was, an interesting observation. And then they also found out the mice also had a decreased sensitivity to pain. And then also some of their learning interactions and their social interactions were also affected. So that raised some questions about, well, maybe what we're learning in the mice might also be something true about this protein in people. And so that raised the question, well, what is BDNF's role in all of this? And that is when we began to invite patients to come to the NIH so that we can learn not only about BDNF, but also to learn about maybe other genes in this whole region that could be important for taking care of patients. And to really get the whole array, we also invited people who only had aniridia. So those are people who have isolated mutations just in PAX6, and then healthy children of similar ages. And we had everyone come to do a really long series of tests. And as Kelly mentioned, it's a week long. Some families stayed longer than a week, and was like, you can see it's like a whole page of different tests. And we're actually still analyzing this data because so much data was generated from these visits. And we had a total of 110 families participate either by coming or by sending samples and filling out questionnaires. So back to BDNF. So this really complicated diagram just shows you all the different hormones that are related to body weight regulation. So it turns out the adipocytes, which are the building blocks of the tissue in our body that make up the subcutaneous and visceral fat in our body, make a hormone called leptin. And leptin acts in the brain to kind of, it's like a thermostat. It tells the body how much storage you have of energy. And from there, it actually sends all kinds of other signals to control appetite and body weight. Well, it turns out that BDNF is downstream of this whole pathway, and that's part of why it has a role in body weight regulation. And so one of the things that we found out was that patients, depending on whether or not they had two copies of BDNF or one copy of BDNF, they have very different body weights. And that's what raised the question that, you know, BDNF may be important in people in addition to the other studies that have been previously done. And so what this graph shows is that everybody who has the yellow bars, those are the people with deletions that were big enough to include BDNF, compared to the people with the blue bars, where there's 2 copies of BDNF intact. You can see that there's a much higher body mass index score for those patients. And then if you kind of just look at it, you know, just kind of a yes/no question, whether or not childhood obesity occurred before the age of 10, we found that all of the patients with BDNF loss had this feature. And whereas people who had both copies intact, pretty similar to the background prevalence of childhood obesity in the United States. And this is really important because then we could actually start to counsel patients. And because of our ability to counsel, we're actually changing this pattern. So families who know in advance, like they can have preparation and make changes even before it happens. And so that's why we're able to change the face of this. So the 100% thing is no longer like a for sure thing anymore. And then the other thing I just wanted to mention is that body mass index is really kind of a weight-based thing, and it doesn't really tell you body composition. So at the NIH, we also looked at percent body fat, muscle mass, and bone composition to make sure that we were really looking at true adiposity. And indeed, having BDNF loss is a risk factor for that. In terms of how that is distributed in the body, it's actually similar in terms of visceral versus subcutaneous. And you know, basically the bottom line is that we feel that it's not that there's anything different, like in terms of how energy is handled in the body. It's really more about hunger. And so that if we can make some changes to nutrition, exercise, and lifestyle, we really can reverse this because there isn't like a fundamental problem with the visceral to subcutaneous ratios. And that's why if we can get that under control, we can also get things like blood pressure and lipids and diabetes risk also under control. So it gives us hope that we can intervene and make a difference. This is showing hunger scores, and these are based off of questionnaires about how hungry people feel. And the people with BDNF loss certainly felt more hungry, and that actually changed how we talk to patients. Because I think if anyone's been involved in kind of really routine sort of kind of weight management, there's a lot of discussion about mindfulness, about eating, and about hunger cues and things like that. Well, that doesn't really work with people who feel hungry all the time. And that kind of advice to a patient may not be helpful if they always feel hungry. So if you tell someone, oh, just eat to the point of no longer being hungry, and that way you won't gain too much weight, that's not helpful if we don't figure out ways to handle that hunger feeling. And so that's led us to want to do studies to look for either specific medications or specific combinations of food or timing of eating that can make changes to hunger in relation to BDNF. And so those are ongoing studies. We also looked at energy expenditure just to make sure that there wasn't a metabolism problem. And the good news is that there isn't. So again, if we can figure out ways to help with the hunger, that it can actually help a lot with the metabolic risks. And then this is just commenting on how, like in mice, remember I showed you that picture of the mouse with the BDNF loss and then its wild-type littermate. Like basically, if we do pair feeding, meaning that we have the mouse eat the same as its brother or sister in the same litter, then we can also prevent the excess weight gain. So it really shows that it's not a metabolism problem, but more so a hunger concern. And then we wanted to make sure that we didn't only focus on just BDNF. So this is actually lining up all of the different deletion boundaries. And we saw that there wasn't really any pattern based on the end of the deletion on this end. But then when we line things up based off of the telomeric end, we can see that there's a pattern as to who develops increased body weight and who doesn't. And it turns out that it actually is at this particular line. So if you have this particular gene affected in your deletion range, then there's a higher risk of gaining excess body weight as a child. And whereas if you had a deletion that stopped before this point, that's where all the patients with normal weight resided. And so it actually did turn out to be BDNF. So our guess was good and didn't identify a separate locus for us to look at. And then the other thing we found out is that you don't have to have the entire gene affected. You can just have part of it deleted, meaning that only a portion of the BDNF gene being affected can actually cause all of the symptoms in terms of the hunger and the weight gain. Because if you have a deletion that ends before just that first little portion of BDNF, then you don't have that propensity for the weight gain. So then that led us to doing studies of looking at why these different transcripts matter in the brain. And we looked at all the different expressions. And this is using tissues from like stored samples and other larger studies at the NIH. And the bottom line is that those first early parts of BDNF are important because they make up a huge part of the BDNF that's expressed in the brain, that is brain region that's important for appetite. And so that's what led us to have mice where we actually did disruptions of different parts of the BDNF gene. And then we found out that there are only certain ones that kind of affect weight, whereas other ones don't affect weight. That's what all these different little bar graphs are showing. And the reason why that's important is because now we can have more targeted approaches for trying to treat this, like because maybe we don't need to alter all of BDNF, but just certain portions of it to make a difference. And so we are basically working on targeting those first exons so that we can leave the other ones as they are, so that we don't affect the other things, which I'm going to mention in a little bit, which is pain perception. So let me just move on to that because I just want to make sure I save enough time. So in terms of pain perception, the reason why BDNF is important in pain is because whenever you like stub your toe, that signal has to go from your toe to your spinal cord and then to your spinal cord to your brain. And it's that relay in the spinal cord that is mediated by BDNF. So that means if you don't have enough BDNF, then you almost have like a muffled signal. Whereas if you have too much BDNF, you can actually cause too much pain. So that's where the challenge gets, where we're trying to figure out a way to control BDNF so that we can help people not feel hungry, not to gain too much weight. But if we do it incorrectly, we might actually cause people to have increased pain. So that's why it's really important as we're like doing this research to make sure that we are targeting the right things to help people feel better. It turns out that the mice also have a difference in pain response, and so they can serve as a good model for studying drugs and treatments. I just want to point out that when they did put in extra BDNF into the spinal cord of mice, that actually did cause differences in the pain response, so that's why we have to be careful. And then what made me interested in all of this is because families have been reporting to me that there was high pain tolerance in some but not all patients with WAGR syndrome, and that kind of led to this line of thinking. And so when we compared people with and without BDNF affected, we found that there was a lower pain response in the people with BDNF affected. And this is really important because pain is actually part of what allows doctors to know if they need to do a workup. So there are some conditions like, say, for example, pancreatitis, which is extremely painful for typical patients, but in people with WAGR, some of them actually weren't really describing much pain. And that could cause us to miss a really important diagnosis and get treatment sooner. Or there were cases of people who had a fracture of a bone and they were still walking on the leg, whereas a typical child feeling typical pain would have been limping and would have gotten an x-ray a lot sooner. So that's why it's important to know if somebody has like a really high pain threshold, because you might miss a pain response. One of the things that we did was we did nerve conduction studies, because treatment for Wilms tumor can affect sensation and nerves. So we wanted to make sure that wasn't the reason for this difference we saw, and everything was normal in this. It's just a bunch of numbers, but just wanted to say that the nerves were fine. But it was the perception. So what that is we had the patients hold hot and cold thermodes to say, okay, how much does this feel? Like, does it really hurt, or it barely hurts, or you don't even notice anything? So that's what those little pictures are showing in terms of patient response. And so for hot pain and cold pain intensity, the patients with the yellow bars, those are the people with BDNF loss, they reported a sense, they could feel it. It's not like they couldn't tell it was hot and cold. It's just that it didn't feel like it hurt. So that was the thing that was very important for us to understand, is that it wasn't, again, a nerve problem or an inability to feel. It is the perception, or kind of the augmentation of the pain perception that was altered in people with BDNF loss. And this is just showing an average score of all of those previous graphs. Next, what we did was we started looking at animal models, because again, we need animal models so that we can test medications and treatments, because we really want to make sure things are safe and that they work in those models first before we even consider trying in people. And so this is just showing you that in the pink bars, those are the rats that have BDNF affected, that you have to have a lot higher temperature, or to be really hot, or really, really cold temperature to be really cold for them to actually feel the same level of discomfort. In this case, it's pulling their paw away or lifting their tail. So it's kind of like physiologic responses that the rats are doing. And again, these are just a bunch of pictures of different animal studies, but just wanted to show you that we do have things going on in the research lab to look for medications and treatments. The other thing that we saw in the mice was that they had differences in learning and social interactions. And so that's what made us interested in BDNF and its role in either autism spectrum disorders or intellectual differences. And it turns out that even common variants in BDNF in just the general population are also can affect cognitive performance. Like they did even studies on like being able to read a map and being able to like navigate in a city after looking at a map, and found that just these common variants, which are common in about a third of the population, that there are different people who have different abilities to be able to read a map and to be able to go around a city. And so even common variants, so not just rare disorders of BDNF, can affect these types of functions. And so that was the original Vanderbilt study that Kelly was talking about, showed all these prevalences of autism like features and intellectual disability, and that's what got us interested in this. And what we found is that people who have BDNF loss do have a lower cognitive function score on IQ testing, as well as on adaptive behavior, which is a Vineland questionnaire about activities of daily living and independence. So that was one thing that we realized was that BDNF was a major factor in this. The one thing, though, to keep in mind is that bigger deletion sizes are typically seen in BDNF, and so we wanted to make sure it wasn't just a larger deletion size, but within groups, like whether or not you had BDNF deleted or not, the size of the deletion didn't really matter. So it really seems as if BDNF is included in the deletion region or not, that's kind of the key feature for the difference in intellectual function. And then with regard to autism, the different components of autism symptoms are social, communication, and repetitive. And it turns out, so the social behaviors were the ones that were kind of the most notable with BDNF, and in kind of pushing the diagnosis of autism spectrum. But when we actually looked at psychologist-based observation scales, the rates of actual autism were similar, suggesting that it's really features of autism and not like a full-blown condition that may be what's more prevalent in BDNF loss. However, I will say that for getting special services and school services, having the label of autism diagnosis is actually helpful in many states in the United States. So our way of approaching this is that we help families like in whatever way to get the most services they can with schooling. So if it means getting that label, even if your child doesn't really meet like full-blown criteria, we actually prefer to just say someone's on the spectrum so that they can get for services, because really our goal is to help everyone achieve their fullest potential. But if one really wants to know, does a child really have autism or not, it's really important to have like a trained neuropsychologist doing testing, because all those questionnaires alone can maybe overdiagnose based off of those social cue differences. So some of the other genes that we became interested in looking at was PAX6. We already knew that was involved in the aniridia, which is the iris, but we were wanting to know if there are other things that we should be paying attention to. So it turns out that PAX6 is really important in the brain and in the pancreas in terms of its formation. And so what we did was we started looking at brain MRIs of patients and actually talking with patients about their symptoms and looking at things to really correlate like symptoms versus what we're seeing on MRI. So here you can see that a person with PAX6 is missing this little circular thing right here. That's the pineal gland. And then you can see here, there's no little circular thing there. So that there's an absence, or at least a very small, so you can barely see it on MRI, that there's pineal hypoplasia, meaning smaller pineal gland. Well, it turns out that the pineal gland is really important for making melatonin. And many of us have probably heard of melatonin and used it to help as a sleep aid. So in people who don't make enough melatonin, you can imagine that might influence sleep, and that sleep in turn affects a lot of other things the next day in terms of behavior, learning, just feeling good in general. So we looked at sleep disturbances and melatonin levels, and we found that by having this hypoplasia, this small pineal gland, and this MRI finding and this PAX6 loss, that all these things can be bundled together, and that sometimes treating with melatonin can help with symptoms. There's another gene called C11 open reading frame 46, and we were interested in understanding whether or not that could affect the corpus callosum, which is the part that connects two halves of the brain. It is important for auditory processing, executive function, various different types of higher order thinking. And you can see that having PAX6 loss can affect the corpus callosum, but having PAX6 and a C11 or 46 can make it even more so. So that's why I think that it's combinations of genes in this region that may be important, and not really even just focusing on a single gene. And this is just giving you a pictorial representation. So you can see that the side, it says that the little kind of rainbow-like thing here, that's the corpus callosum. You can see how different it looks when you go from all the genes are fine to PAX6 affected to PAX6 and C11 orf for both males and females. You can see the difference, how much smaller it is here. So if we do have right-left hemisphere connectivity differences, what can that do? So hearing loss is similar in patients with WAGR syndrome or aniridia. And when we looked at auditory processing, though, so that's not just the ability to hear, but how it travels within the brain, that there are some differences that then occur because of the effect of PAX6 and the C11 or 46, and it is an additional piece that can make it even more so. And then there was other findings that we found about PAX6, which is that patients were sometimes being reported as, oh, well, you know, there was some clumsiness, maybe they were bumping into things, and people just assumed it was visual and neurocognitive. But when we really looked at it, it turned out that patients were having double vision when they were turning their head from side to side. When they tried to look to the edge, they would actually suddenly start to see double. And so when we asked people to do this while we took pictures, that's when we found out this child is being asked to look this way. And you can see that this eye is moving along like it should, but this one's getting stuck in the middle. And so that basically means that instead of having the eyes conjugated together and being yoked together to be able to see things clearly, you're going to end up with double vision because they're not lined up right. And it turns out that when we looked at the brain, this little black line represents cranial nerve 6. Well, it's missing on this side. And so that explains why there was a nerve palsy or a nerve problem that was preventing the eye movements. And so that's another thing that can be associated. That's just showing you the nerve and the missing nerve in that picture. The other thing that we noticed is that patients also tended to have shorter stature if they were having bigger deletion sizes. And so we're still collecting and understanding all the different genes that affect height. And it seems as if it's an accumulation of lots of different small regions. So it's not one specific gene, but rather the total number of genes. And again, that's something we're also studying. Other things that we've been looking at are things like palate abnormalities, like narrow palates, sort of flattened facial positioning that can affect breathing at night, such as obstructive sleep apnea. All of these things, there are different genes that may be affecting each one of these things as well. And then the other thing that we found out is that WT1 can also affect Mullerian regression. And what that means is when a baby is forming in the womb, the uterus is actually something that is kind of generic default on. So female is like the default patterning of development. And then you have to have certain genes that tell the uterus to not be made for a male patient, for a boy, for a baby boy to properly develop in utero. And so if you have some WT1 problems, it's important to know that sometimes you can have a remnant of the Mullerian duct, which is a remnant of what would have become the uterus. And so what happens is it can serve as a pouch that collects bacteria and recurrent urinary tract infections. So in particular, if a male patient like has recurrent urinary tract infections, it's really important to image and look and make sure that there isn't this going on, because if it is, then that is something to consider as to whether or not you need prophylactic antibiotics, or whether or not one might want to do a laparoscopic procedure just to clear out that extra remnant so that you don't have like a nidus for infection. And then we also learned some practical things. So not necessarily things that are like, going back to the research lab to figure out a drug, but rather like, let's talk about practical things. So our low vision occupational therapist talked a lot with patients and families about how to make schooling better, and also talked about adaptive devices to help people have more independence and how to deal with auditory processing disorders. So I just kind of wanted to share this slide real quick, which is the contrast that patients can see when they have aniridia. So for me, I can see all the way down to the letter C. Well, actually, no, I can see the letter V here. I don't know how well other people can see it, but in patients with aniridia, they usually report that it's kind of up here is where the contrast difference is, like they can't see the letters anymore. And so it's really important to know that, because if in the school you're using paper that's kind of like this, like very pale lines, like either very faint blue lines or like it's blue paper with little white lines, no one's going to see that. They're going to be like writing like all over the place, because you need to have really clear lines. And so just kind of basic just basic knowledge about this, like really can make a difference for a child to feel confident in school and to be able to good handwriting skills and things like that. And then the other thing is that yellow tinted glasses can help with the contrast and reduce glare. And so these are some things to think about when a child with aniridia is experiencing some visual differences. Dexterity, there are different things that you can do. If I think a lot of the kids don't like to have the Velcro kind of shoes once they get to a certain age, and so they want to have the tie shoes and they like the design. So one of the things that we found is using these coil shoelaces can be helpful because you just pull on it to tighten and it snaps back and it keeps things in place. So you don't have to actually tie the bunny ears and do all of that, but you can still use lace shoes comfortably, and it's really fun, and you can have different colors and it's attractive. So like I think for the kids, that was something they really liked. We also handed out bowls that had folded edges so that food wouldn't come off, and then these special gripping spoons with these wraps around the hand so that with dexterity concerns, with people with lower muscle tone, for example, that we could have people have more independence using these devices. And then for auditory processing, using FM devices where you place an amplifier near the teacher, in addition to having the child toward the front of the room, and then wearing an earpiece that really amplifies the signal to noise ratio, meaning that all the other kids are talking, or like if there's a lot of background noise, if you have the teacher's voice really close to a speaker and it's really funneled to the patient, then they can actually hear that better than other things. Because otherwise, most of us are able to filter out background noise or have right left ear, like be able to hear two things going on simultaneously. But in patients with auditory processing disorders, it's really hard. And so that's why amplifying the signal that you want them to hear is really critical. Some of the ongoing studies, so thank you for those of you who did send teeth to Dr. Larry Ryder. He's still doing the research at UTHSC and University of Tennessee. And our hope is to get some funding for the NIH to do more studies with those cells. Meanwhile, I'm working on using pluripotent stem cells taken from blood samples, and we're differentiating them into neurons, and we're using them to look for different drugs to help with BDNF augmentation and TrkE receptor activation. And then I'll be emailing a lot of families to give me updates because Dr. Andrew Murphy at St. Jude has been really looking into Wilms tumor risk because of our recent concerns about the age range and just that we're learning that a lot of the mantras that we've had in the past are not correct, and that we need to be more aware of these things. And so what we're trying to do is to have a longer scope and more genetic detail to really understand Wilms tumor risk. And I think that's it. So I'm done. This is just a thank you to all the collaborators, an extra special thank you to all the research participants who participated, and thank you in advance. I'll be emailing people to get updates to update our registry. And then I just really wanted to say how much I appreciate Sherry and Amy is so dear to me. And I think that the reason for me even doing research in the first place is really because of patients, and I'm just so grateful for the privilege of being able to take care of patients with WAGR syndrome. So thank you so much. And this is my contact information. Call, you can text me. I'm a pediatric endocrinologist by training, and so if I can't be helpful, I can at least find a colleague who can be, and so I would love to be helpful in any way that I can to anyone in need. So thank you so much. I'm also happy to answer any questions.